thought by a pen. And وعلم الإنسان ما congratulations to all of my dear students for the holy month of Ramadan. And I am sure you all are okay and right and you are doing well. Today topic is membrane potential and action potential in smooth muscle. My name is Professor Dr. Taj Muhammad Khan, and I am working in Swat Medical College as head department of physiology. I am sure that you all know that the concept of action potential. There are certain general principles how the action potential is generated and how the action potential is then spreads, propagated along the membrane of the muscle membrane surrounding the muscle fiber. And you know that how the action potential causes contraction. The creation, generation of action potential and contraction, these are the two different things. First, there is generation of action potential, and then that action potential is responsible. That action potential is responsible for the contraction of different muscle or nerve fiber. You have studied in the skeletal muscle how the action potential is generated. First, briefly, I'm going to discuss the basic concept, and here you can see the concentration of the different important ions or chemical substances across the membrane that surrounding the muscle fiber or the nerve fiber. You can see the most important, the electrolyte, that is the sodium. You can see the concentration of the sodium at outer side of the membrane, that is 142. And you can see that the concentration inside the membrane, that is 10. And the concentration outer side is more than 14 times greater. And here you can see that the concentration of potassium at outer side of the membrane, that is 4 milliequivalent per liter, while the concentration of the same ion, the potassium ion, that is 140 milliequivalent per liter, and that is 35 times greater at the inner aspect as compared to the outer aspect. And here you can see the calcium, the concentration of the calcium at the inner side is more than 10,000 times greater as compared to the outer side. So this is just a brief. We are not going in detail, but your concept about the concentration of the different important 3-4 ions, I want to that concept is clear. Now we are going how the action potential it is generated. First of all, you know that one is the all the terminologies used in physiology having their own definition and as well as explanation. First of all, the membrane at resting condition, you know, it is having a potential. That potential is called resting membrane potential, and you can easily define that the resting membrane potential of the membrane at rest, and then how that. Hello.
بس the आवाज ډېر زیات سلو دی سره لږ د مایک د ځای کې سره سلو دی
How go stick there?
आवाज کلیر شو سره
Hello, clear, Shekha. You know that the resting membrane potential, that is the potential of the membrane during at resting condition, and how that resting membrane potential is converted into the action potential. By definition, the action potential is the potential of the membrane during action. I am saying that the terminologies having their own definition and as well as explanation. For example, conductive system of the heart. You can easily define that it is a specialized system present inside the heart responsible for the generation and as well as for the transmission of impulse all over the heart. So here, how the action potential is generated. You know that when there are certain stimuli, they are responsible for the bringing the change in the permeability of the membrane. For example, electrical potential. Some hormones they are acting, and some neurotransmitter, the transmitter. These are the chemical substances. They are released by the free neuroendings, and some other substances can excite, can bring the changes in the permeability of the membrane. Once when there is electrical excitation of the membrane, you know that the concentration of the sodium that is at outer side is 142 milliequivalent per liter as compared to the inner. The membrane surrounding the muscle fiber or the nerve fiber is highly supplied with the voltage gated sodium channel. In case of the skeletal muscle, only the voltage gated sodium channels they are present. And that is gifted by the God. That is to the human being. And we are discussing the smooth muscle fiber and first how the action potential is generated. There are two types of action potential. One is the spike action potential. The second one is the action potential with plate. In the skeletal muscle, only the spike action potential is generated, while in case of the cardiac muscle fiber, in the smooth muscle fiber, the spike action potential and plus in some of the fibers, the action potential with the plateau. When there is any electrical excitation of the membrane, just briefly, I am going quickly to cover the topic, which is allotted to me. When there is a application of the electrical stimulus onto the membrane, you can see the main action of that electrical stimulus that is on the voltage gated sodium channel. And what is the action? The voltage gated sodium channels uh, open ho jate, they are open, and you can see that the concentration of the sodium ion is outer 14 times greater. So due to the concentration gradient difference, that is 14 times greater as compared to the inner side. So the sodium ions, they are transmitted from the exterior towards the interior. And you know that the sodium are the positive ions. And you can say that that is the transfer of positivity from the exterior towards the interior. And you know, it is a confirmed principle that in the large nerve and muscle fiber, the resting membrane potential is minus 80 to minus 90 MV, millivolt, right? In case of the small nerve and muscle fiber, the resting membrane potential is from minus 50 up to minus 60 millivolt. So when there is a transfer of positivity from the exterior towards the interior, at resting inside is negatively charged. So that is the transfer of positivity from the exterior towards the interior. For each nerve and muscle fiber, there is a limit. We can say that is the threshold for excitation. When that change, for example, minus 90 inside the muscle fiber, when there is transfer of positivity, so that minus 90 changes into minus 80, into minus 70, into minus 60, into minus 50. If the threshold for excitation, just assuming for small nerve fiber or muscle fiber or for the large muscle fiber, if we are just assuming that the threshold for excitation is minus 30. When that change in positive direction, due to the diffusion of sodium ion by the simple diffusion. What is simple diffusion? Simple diffusion by definition, the transportation of the chemical substances from the higher concentration state towards the lower. That is energy independent process. That is a downhill process. So the positivity is converted. If that change in positive direction, if that crosses that limit, just assuming minus 30 MB, if that limit is crossed, so you can say that the membrane is depolarized. The membrane is depolarized. At resting, remember at resting, memorize that the resting membrane potential is at the membrane is polarized at resting. Memorize. Polarized means that two sides, two poles. One is positively charged and the second one is negatively charged. Outer side is positively charged and inner side is negatively charged. So polarized, membrane is polarized, and due to the transfer of positivity from the exterior towards the interior, that is the positive change. If that change crosses that limit, which we are saying that is the threshold for excitation, then you can say that the membrane is depolarized. Depolarized just मैंने कहा कि terminology में definition है जो पोलराइज कंडीशन था उसको ब्रेक किया, डीपोलराइज किया, उसको डिफॉर्म किया, उसकी ब्रेकिंग डाउन फ्रॉम द पोलराइज इनटू डीपोलराइजेशन. Not complicated, just simple. That is just the breaking down of the polarized condition. And that is due to the influx of the sodium ion, the positive ions through the voltage gated sodium channels, you know. And these are the channel voltage gated, they are called the fast channel, because these channel they open within a short period of time, over 10,000 times of a single second. And that when there is the membrane is depolarized, there is immediately closure of that sodium voltage gated sodium channel. And here you can see that graphically, this is just, you can see here, from here you can see, if that is plus positive, the threshold for excitation, if this limit crosses that limit, which we are saying the threshold for excitation, then you can see that the membrane is depolarized, that is the spike. And immediately after the closure, within 10,000 of a single second time, a shorter duration of time, that is immediately followed by the opening of the voltage gated potassium channel. Here you can see the potassium. And you know that the potassium ions concentration inside of the membrane is 35 times greater. So due to that concentration gradient difference, so the potassium ions that is transmitted, that is transported from the interior towards the exterior. And here in this graphical presentation, you can see that this is now, this is further creation of negativity inside the membrane, because there is transfer of positive ions from the interior towards the exterior. When that change in negative direction means the creation of negativity, once again inside the membrane, you can see when it reaches to that minus 60 or small muscle or nerve fiber, yeah, minus 90 in large muscle fiber, then that is the repolarization, rebuilding of the polarized condition. Yani rebuilding of the polarized condition. Clear? At resting, the membrane is polarized. During action, transfer of positivity from the exterior towards the interior, and then closure of voltage gated sodium channels, then the immediately opening of the voltage gated potassium channel, and the concentration inside the membrane of the potassium ion, that is 35 times, you can see 35 times greater. So once again there is diffusion of the potassium ion from, and that is the change inside the membrane in negative direction. If that change in the negative direction, that reaches to that limit in large muscle and nerve fiber, that is minus 90 MV millivolt, and in small nerve and muscle fiber, that is minus 50 up to minus 60. If that achieves that limit, then you can say that that is the repolarization, rebuilding of the polarized condition. So that is the action potential, spike action potential. And the second one you can see here, one is, this is in the skeletal muscle, only the spike action potential is generated, while in case of the smooth muscle fiber, in some of the fibers, the spike action potential is generated, while in other fibers, here the action potential with the plateau is generated. Here you can see that is the graphic presentation. The action potential in millivolt from Purkinje, any from the nerve fiber, from the muscle fiber, the how the action potential with the plateau it is generated. You can see that the small smooth muscle fibers and cardiac muscle fibers, they are highly supplied with the voltage gated calcium channel, with the voltage gated calcium. So the voltage gated sodium channels are also present, but those channels, they are less in number. And all the channels, memorize all the channels, they are protein in nature. They are provided by the structural protein two types, structure and integral, yeah, globular protein. So all the channels, their walls, they are provided by the protein. So the smooth muscle fibers, these are extremely small muscle fiber as compared to the skeletal muscle fiber. So we are going to discuss the action potential with the Plato. Like just I want to show you that here you can see that the smooth muscle fibers, they are smaller in diameter and smaller in length, 1 to 5 micrometer in diameter and about 20 up to 500 micrometer in length. And the skeletal muscle fiber, that is about 30 times greater in diameter as compared to the skeletal muscle to the smooth muscle fiber, while that is up to 1,000 times that is greater in length. And the smooth muscle fibers, they are of two types. You can see here, this is the multi-unit smooth muscle fibers and the unitary smooth muscle fiber. And the you can see what is the difference. So this is the complex. You can see that this one is the complex of the small fibers. This is the smooth muscle, and each muscle is formed by the combination of the different muscle fiber. In this case, in case of multi-unit, their definition is present inside their terminology. Multi-unit smooth muscle means that is complex of the small size muscle fibers. And these muscle fibers, you can see that is, these are the discrete separate muscle fibers, and these are the muscle fibers. They are connected at different spots. And these muscle fibers, each muscle fiber is supplied by a narrow fiber. And you know that this is in case of the multi-unit smooth muscle fiber. These are the muscle fibers. They are usually, they present in the ciliary muscle of the eye. They are present in the muscle muscle of the iris of the eye, and they are present in the piloerector muscle of the hair. You know that these muscles, each fiber is supplied by a narrow fiber, free narrow ending, form a neuromuscular junction, and each muscle fiber, it functions individually, not combinedly. Individually, on the other side, this is the unitary smooth muscle. We can say that is the visceral smooth muscle, because these muscle fibers, they are present in the gastrointestinal tract, in the uterus, in the ureter, and most of the blood vessels. And so we can say this is the visceral smooth muscle fiber. And you can also say the syncytial smooth muscle fiber. The reason is that it acts, just like present in the form of sheet or bundle. It is present in the form of sheet and bundle. And these muscle fibers, they are adherent to one another. And a large number of the voltage gated channels, they are present. And those are the channel, sorry, the gated channels, they are present. And those are the channels through which the ions can move from the one muscle fiber to the next. And that is the main function of this is, once it acts as a unit, it acts as a unit, as a sensation, as a unit, especially in the cardiac muscle, in the smooth muscle fiber, and most of the visceral organ. These are the, especially in the GIT, uterus, and the blood vessels. The smooth muscle fibers, they are present in this form, in the form of sheet or in the form of column, right? A main function वहाँ पर individual muscle fibers, they are excited individually, not combinedly, but here it acts as a unit. If there is generation of action potential in any fiber of that column of that sheet, so that action potential is at the same time that is transmitted from muscle to muscle, and that is transmitted to the whole sheet, the whole column. So we are going back that how here you can also see that one is the, in case of the smooth muscle fiber, today's topic is the membrane potential. Membrane potential is mainly means the membrane, the potential of the membrane at rest. Certain factors, they are responsible to generate the resting membrane potential and to maintain resting membrane potential. You know that the membrane surrounding the muscle fiber, that is also highly supplied by the potassium sodium leak channel, yeah, leaky channels. And you know that at resting condition, the concentration inside of the sodium is greater as compared to to the outer side about 35 times greater. So you can see that at resting condition, there is leakage, there is transmission of the potassium ion through the leaky channel from the interior towards the exterior. That is leaking of the positivity. So that plays a main role in the creation of resting membrane potential. So that is the transfer of positivity from the interior towards the exterior. The second role that is played by the sodium potassium bond. So the membrane potential means that the potential of the membrane at resting in case of the small smooth muscle fiber, that is minus 50 up to minus 60 millivolt, right? Now we are discussing, that is just graphic presentation of how the action potential, spike action potential, that is generated in the skeletal muscle. Only the spike action potential is generated. And later on, that action potential, that is spreading, and that is transmitted along the membrane surrounding the muscle fiber. Here you can see that is the transmission of the action potential. And then there is another terminology, the excitation contraction coupling mechanism. And there is generation of action potential. You can see that the membrane is excited. That is the generation of excitation. That is the generation of impulse. That is the generation of electrical potential. And that generation of action potential, then that spreads along the membrane surrounding the muscle fiber, and that then causes the contraction of the different muscle fibers. Now we are discussing just that is how the action potential with the plateau is created, is generated. This is just a graphic presentation. You can see here. So here in case of the smooth muscle fiber and as well as in the cardiac muscle fibers, memorize the mechanism is the same. The membrane surrounding the muscle fiber in case of the smooth muscle and as well as in case of the cardiac muscle fiber, they are highly supplied by the voltage gated calcium channel that is gifted by the guard. And there is need for the prolonged action. So the muscle fiber, and the cardiac muscle fiber, and as well as the smooth muscle fiber, the membrane surrounding these fibers are highly supplied, are highly gifted by the guard voltage gated calcium channels. The sodium channels, they are also present, but they are present less number of sodium channels, they are present in the membrane. Once there is a application of the electrical potential on the membrane, first there is opening of the voltage gated sodium channel, and you know that the voltage gated sodium channels, these are the fast channels. They open and then close within 10,000 times of a single second. And in that case, in case of the spike potential, that is immediately followed by the opening of the potassium, voltage gated potassium channel. But here the situation is slightly different. So the voltage gated sodium channels, they are opened and there is transmission of sodium from the exterior towards the interior. But that depolarization, here you can see, sorry, here you can see that the depolarization is immediately followed by the repolarization. But here the situation is different, that the depolarization is not immediately followed by this repolarization, but there is formation of the plateau. And that plateau is formed once there is transmission of sodium ions towards the interior. Immediately the voltage gated sodium channels, they are closed, and that closure of the sodium channel here in case of the cardiac muscle fiber, in case of the smooth muscle fibers, that is followed by the opening of the voltage gated calcium channel. They are opened by the voltage gated calcium channel. So you can see that the voltage gated calcium channels, these are the chain, the concentration at the inner side more than 10,000 times greater. But these channels, the voltage gated calcium channels, these are the slow channels. Means they open in a larger time, duration of time, and as well as they close in the longer duration of time. So you can see that there is creation of negativity from the interior, inside the interior, and there is, but the channels they open for, they are slow, so they open in longer duration of time and they close in longer duration of time. So there is, due to that, there is formation of the plateau. Importance of that, then the calcium ion they are closed, then they are immediately followed by the opening of the voltage gated potassium channel and the repolarization. The opening of the potassium channel means that is the transfer of potassium positivity from the interior towards the exterior. So the action potential here, the action potential with the plateau in some of the smooth muscle fibers. There is contribution of the three types of voltage gated channels. The sodium, voltage gated sodium channels, and the plateau formation due to the opening of the voltage gated. These are the slow channels, voltage gated calcium channels, and then there is involvement of opening of the voltage gated potassium channel. So this is, so there is depolarization that is followed by the plateau. Then that plateau is followed by the repolarization due to the opening of the potassium ion. A third one is the a third one is that some of the smooth muscle fibers they are extremely smaller. So there is no generation of proper action potential, and those fibers we can say those fibers they are present in the arterioles, they are present in the neat arterioles, and they are present in the precapillary sphincters. So these are extremely smaller, but these fibers they are self-excited. They are self-excited to memorize. They are self-excited. You know, means that these fibers they are smaller in size. They are unable to generate the action potential. They generate by itself. They are self-excited. The depolarization, and that depolarization is not enough strong to create action potential or to transmit or to cause the contraction. So these when they these the that is called slow wave rhythm, slow wave rhythm. The reason is that, and that slow wave rhythm that is occur inside the smallest smooth muscle fibers present inside the arterioles, because the arterioles and the precapillary sphincters and the mid arterioles less, or they are not supplied by the neurofibers. So there is no release of the neurotransmitter, release of the chemical substances. So if that change, so it form the muscle bulk, muscle, muscle mass, you can see, and especially in those structure which I have mentioned. So if that slow wave rhythm, if that is too strong, means that is the transfer of positivity towards the interior, and that is self-excited. That is the generation that is excite. You cannot say the excitation. If there is change in that positive direction, just we are just assuming in case of the small smooth muscle fiber, that threshold for excitation is minus 35, right? If that change crosses that minus 35, then there is generation of action. And this slow wave rhythm, it is just a local property of the muscle. And this rhythm, yeah, depolarization is unable to pass along the membrane. When that change, that slow wave rhythm, that crosses that limit, that is called threshold for excitation. Then there is generation of action potential, and same is the mechanism that the action potential is generated and that action potential is transmitted along the membrane and then cause the contraction. The topic is just membrane potential and action potential in the smooth muscle fiber. And I want to slightly discuss that how the action potential causes the contraction. There is a very well-known mechanism that is called excitation, contraction, coupling mechanism. Excitation, contraction, coupling mechanism. This is here you can see. This is excitation, contraction, coupling mechanism. The topic is huge topic. It needs a month to discuss with you, to explain to you, to clear your concept. But here the time is too short. I am just going. how the action potential causes the contraction. For that, there is a standard mechanism, and that mechanism you can see the excitation coupling mechanism. This is, you can see that this is a portion of the muscle fiber, either the skeletal or the cardiac or the smooth muscle fiber. This is the membrane surrounding the muscle fiber, and you can see that this one is the, it runs transversely. It runs transversely. We are saying these are the transverse tubules. And here you can see these are the organelle of the muscle fiber, that is called endoplasmic reticulum in case of the cell. But here sarcoplasmic reticulum in case of the muscle. Sarcoplasmic reticulum. In case of the cell, endoplasmic reticulum. And here you can see that the endoplasmic, sarcoplasmic reticulum having two portions. The longitudinally running tubular form and the two cisternae, the globular structure at the end. And you can see that the transverse tubules are connected to this. And here you can see that this is, you have heard the name of actin and myosin. The actin and myosin, these are the complexes of the structural proteins. And these are the actin and myosin. We are saying these are the contractile units, because these are the actin and myosin. They interact and they are responsible for the contraction of the muscle fiber. How the action potential, you can see it causes the contraction, and we are saying excitation. You know that the excitation and generation of action potential, the impulse, all are same. Confuse होने की कोई जरूरत नहीं है, same है, different names है. So once there is generation, you know that how the action potential is generated. You can see here that the action is the potential is generated, and that action potential is this in spreading along the membrane surrounding the muscle fiber. And you can see that this transverse tubule, that is just इसकी जो boundaries है, that is just the invagination of the hello. That is just the inward extension of the membrane surrounding the muscle fiber. And here you can see briefly that there is also extension of the action potential through the transverse tubule and to the sarcoplasmic reticulum, and memorize that the concentration of the calcium ion inside the sarcoplasmic reticulum 1,000 times greater as compared to the inner of the cell. So there are a large number of the voltage gated calcium channels. They are present, and due to that, due to the concentration gradient difference, there is a release of the calcium ion, you can see here. And there is also need for the energy, because that the contraction is an active process. There is need for the energy. The ATP is responsible for the provision of energy, and ATPase act on the ATP, and they convert the ATPs into ADP. And finally, so this, the actin and myosin, the myosin structure wise, I want to slightly, this is, this one is, you can see, this is the myosin. Myosin, you can see, 6 chains, 4 small chains, and 4 small chains, and 2 long chains. You can see the more major portion of these chains, the longer chains, they are unfolding around spirally around one another and form the tails. And here you can see the free ends, they are unfolded into globular structure. This is called the head, and the different tails in each skeletal muscle, in muscle fiber, about 200 muscle fibers, they are responsible for the formation of up to 200 muscle fibers. They form the muscle. So the different tails, they bind with one another and form the body. And here you can see that this is an outward extension. You can see the head, the arm, and the body. And the two places, two spots here, we are saying these are the hinges. Hinges means these structure, the outward structure. These are the bridges composed of the head, the arm, and so these, they are hinges means they are flexible, they are able to move. So this, you can see, this is the actin and this is the myosin Once again, I'm going to discuss you. This is the diagrammatic presentation of the actin. Actin is basically composed of the three main components. That is the F-actin consists of the G-actin and the tropomycin and troponin. The troponin you can see here. This is the troponin is the complex of the three proteins molecules. One is the troponin I, that is specific for the actin. The second one is the troponin T, that is specific for the tropomycin, and the third one is the troponin C, that is the active site of the actin. So, in resting condition, you can see the F-actin, yeah, the G-actin, that is present, spirally, they are present around one another with the, you can see with the F-actin. And these, the main function of the tropomycin is at resting condition. It covered that is covered by this tropomycin, the active site. Once there is a release of, you can see this calcium ions. Those calcium ions, they come in contact with the active site of the actin, and there is generation of the attractive forces. And then there are two theories. One is the walk along theory. The second one is the sliding theory. And I think you are having the concept of that one. This is the walk along theory. Once there is attachment of the calcium with the active site, so there is attachment of the head of the cross Brzezinski head का attachment at the two sides. In the next step, the attractive forces they are provided, and there is bending, yeah, tilting in forward direction of the head, and that tilting, that is the moment in the forward direction, and that moment drag that active site of the actin. That is the walk along theory. हाथ में हाथ डाल के चलना, एक साथ चलना, walking in company. The actin and the myosin, they are working, they are contracted in company, right? The second one mechanism, that is you can see the one was the walk along theory, the second one is the sliding theory. This is the relaxed form of the muscle fiber. You can see these are the Z lines, and you can see the concentration of the muscle fiber of the actin and myosin, 500, 1500 actin in each muscle fiber and 3000. That is the ratio is 1:2. And you can see these are the Z lines. They run transversely just like Z alphabet of the, you know, and the actin, they are attached to the Z lines. And here you can see the myosin filaments, they are present here in resting condition. There is minimal overlapping of the end of the actin filaments and overlapping with the myosin. But in case of contraction, you can see that the Z lines, there is moment of the Z lines, and inward moment, and you can see there is approximation, they become near to each other. And there is more overlapping of the actin filaments with each other, and there is over. So that is, there is sliding of these filaments. So we are saying that that is the sliding theory. And that is the sliding theory. You know that this is also a diagrammatic presentation of the tubular sarcoplasmic reticulum and the muscle fiber, a portion of the muscle fiber. And here you can see that is the how the action potential is responsible. Yeah, how the excitation, contraction, coupling mechanism. You are having the knowledge. Just briefly to clear your concept, yani how the action potential that causes the contraction. The action potential generation and the contraction, both are the different things. First, there is generation of the action potential, and then that action potential is responsible, yeah, causing the contraction, right? And there are hello, there are certain terminology. One is when there is a change in the positive direction. Yes, hello. In case of the depolarization, when there is a change in the positive direction due to the influx of the sodium ion, if that change crosses that limit, which is threshold for excitation, just assume minus 30. So then we can say that the membrane is depolarized. action potential is generated. If that change is less than that, there is confusion about the terminology. Then you can say that the membrane is not depolarized. There is no generation of action potential. The membrane is facilitated. The membrane is facilitated. So that is the difference between the facilitation and action potential or the depolarization. Once that change crosses in positive direction, that limit, then we are saying that is the depolarization, the generation of action potential. If that change is in positive direction, yani minus 60, then minus 50 millivolt, then minus 40, and up to that limit, then minus 35. We are assuming, just assume minus 30. If less than minus 35, that change, we are saying that the membrane is facilitated, not depolarized. One is the relaxation of inhibition. In some of the condition, what is the difference between relaxation and inhibition? Both are the same. In that condition, there is a transfer of negativity towards the interior, minus 90. Inside agar resting membrane potential and large muscle fiber or narrow fiber में minus 90 है. So you can see when there is, and that is due to the opening of the chloride ion, chloride channels. Chloride channels, you can see the concentration of the chloride ions outer side. This is the chloride ion. That is about more than 25 times greater at the outer side. In that condition, for the inhibition, there is opening of the voltage gated chloride channel. So that there is, due to that concentration gradient difference, this is, the chloride is the negatively charged ion. So that is the transfer of negativity towards the interior. That is minus 90 agar that minus 90 will be converted into minus 95, minus 100. Then you can say that is called inhibition. Inhibition, right? So these are different terminology. Take 2 minutes के लिए आप rest लें, फिर आप लोगों को मैं invite करूंगा if you are having any question about the topic.